Good morning, everyone, and welcome. I am delighted to have you here today. My name is Dr. S.K. Singh, and I hold a PhD in this field. Today, we will be exploring sterility test method validation. The focus will be on a scientific approach to ensure accuracy, sensitivity, and product suitability. Over the course of this presentation, we will look at the key aspects that support reliable sterility testing. I look forward to sharing insights and engaging with you all. Let's get started. Sterility testing is a crucial process that confirms our products are free from viable microorganisms. This step is essential for ensuring safety and efficacy, especially for products like parenterals, ophthalmic preparations, biologics, and tissue products. These types of products require strict sterility assurance because they are often administered directly into the body or come into contact with vulnerable tissues. The standards and guidelines governing sterility testing are set by various authorities, including the USB, EP, JP, WHO, and PICS. Each of these organizations provides specific protocols to ensure consistency and reliability in testing. It's important to remember that sterility is a critical quality attribute. It directly impacts patient safety. If a product isn't sterile, it could cause infections or other serious health issues. That's why rigorous testing and adherence to these guidelines are so vital in our industry. Overall, sterility testing is a fundamental part of our quality assurance process, helping us deliver safe, effective products to patients worldwide. Now, let's talk about why it's so important to validate your sterility test method. First, there are regulatory requirements from agencies like the FDA, EMA, and WHO that you need to meet. Ensuring your method aligns with these standards is crucial for compliance. Next, validation helps confirm that the method is suitable for the specific product you're testing. Different products can have different characteristics, so your testing method needs to be appropriate for that particular type. Another key point is the method's ability to detect even very low levels of microorganisms. This sensitivity is vital to ensure the safety and sterility of your product. It's also important that the method doesn't get interfered with by the product formulation itself. Some products can contain ingredients that might mask microorganisms or affect the test results, so validation helps verify that this isn't a problem. Finally, when you validate your sterility test method properly, it builds confidence. It reassures everyone involved, whether regulators, quality teams, or management, that your routine sterility testing is reliable and accurate. All of these points come together to emphasize why validation isn't just a regulatory box to check. It's a core part of ensuring product safety and quality. Let's take a closer look at the two main compendial methods used in sterility testing. The first is membrane filtration, which is preferred for filterable products, because it allows us to trap microorganisms on a filter and then incubate them. The second method is direct inoculation, which is typically used for non-filterable products, where we directly introduce the sample into the growth media. Now, regarding the incubation media, we have two options. The first is fluid thioglycolate medium, or FTM, which supports the growth of both anaerobic and aerobic microorganisms. This makes it versatile for detecting a wide range of microbes. The second is soybean casein digest medium, also known as SCDM or TSB, which is primarily used for aerobic microorganisms, those that need oxygen to grow. The incubation period for these tests is generally 14 days. This duration ensures we have enough time to detect any viable microorganisms that might be present. The overall aim of this process is to detect any viable microorganism, no matter how small or slow-growing, to confirm the sterility of the product. In summary, these principles form the backbone of sterility testing, helping us ensure that the products are free from microbial contamination before they reach the end user. Let's talk about some key components of method validation, starting with specificity and suitability. These are crucial because they ensure our method accurately measures what it's supposed to, even in complex samples. Sensitivity is next. It tells us the smallest amount of analyte we can reliably detect, which is vital for quality control. Then we have the limit of detection, or LOD, which defines that minimum detectable level. Knowing this helps us understand the method's limits. Robustness is also important. It assesses how consistent our results are under slight variations in conditions, ensuring reliability. Method suitability testing, or MSD, looks at whether the method is appropriate for the specific product, including checking for any inhibitory effects that might interfere with the analysis. Environmental and operator qualification ensures that the testing environment and personnel are qualified, maintaining high standards of accuracy. Media growth promotion testing, or GPT, is used to verify that microbiological media support the growth of microorganisms, which is essential for valid test results. Finally, all these components, specificity, sensitivity, LOD, robustness, MSD, qualification, and GPT, together form the foundation of comprehensive method validation. They help us ensure our methods are accurate, reliable, and fit for purpose, supporting the overall quality and safety of our products. In this slide, I want to walk you through our main objective, which is to demonstrate that our method can effectively detect a wide range of microorganisms, even when they are present alongside the product. This is crucial because in real-world scenarios, contaminants are often found mixed with the product itself, so our detection method needs to be robust and versatile. Now, moving on to our approach, we faced a significant challenge with standard test organisms. These include bacteria like Staphylococcus aureus, Pseudomonas aeruginosa, Bacillus subtilis, and Clostridium sporogenes, as well as fungi such as Candida albicans and Aspergillus brasiliensis. These organisms are commonly used as benchmarks because they represent different types of microorganisms that could potentially contaminate products. Our goal was to ensure that our method could detect these microorganisms reliably, and specifically, that we could observe detectable growth in both types of media we tested. This acceptance criterion is important because it confirms that our detection system is working properly across different conditions and sample types. Lastly, I want to highlight the importance of specificity and selectivity in this context. Essentially, we need our method to accurately identify these microorganisms without false positives or negatives, even in the presence of complex product matrices. Achieving high specificity and selectivity ensures that our detection is both accurate and reliable, which is key for quality control and safety assurance. Now, let's talk about the purpose of this step. Essentially, we want to confirm that our product does not inhibit microbial growth. This is crucial because if the product were to prevent microbes from growing, it could interfere with our testing results and lead to inaccurate conclusions. Moving on to the procedure, we inoculate the product with less than 100 colony-forming units, or CFU, of the test organisms. The idea is to introduce a small, controlled amount of microbes and see how they grow in the presence of the product. Then, we compare this growth to a control sample, one that doesn't contain the product at all. The key here is to look for recovery that is comparable to the control. If the microbes grow just as well as in the control, it indicates that the product isn't inhibiting their growth. The acceptance criterion is straightforward. Growth must be visible within 14 days, and it should be similar to what we see in the control. If we observe this, we can confidently say the product doesn't interfere with microbial growth. This step, known as the method suitability or product interference test, is really about ensuring our testing environment remains accurate and reliable, so we can make informed decisions about the product's safety and effectiveness. On this slide, we're focusing on the lowest number of microorganisms that can be reliably detected, which is a key aspect of method sensitivity. Typically, this is demonstrated during method suitability testing to ensure our detection methods are working properly. The LOD target, or limit of detection, usually falls between 1 and 10 colony-forming units, or CFUs, depending on the method and the sample. This range indicates the test's detection capability at very low levels, which is crucial for accurate microbial monitoring. When a test can detect such low levels, it demonstrates that the test is highly sensitive, meaning it can identify even small amounts of microorganisms. This high sensitivity is important because it helps us ensure safety and quality control in our processes. So, in summary, understanding and validating the LOD helps us confirm that our testing methods are capable of detecting microorganisms at the lowest possible levels, which is essential for reliable results and overall quality assurance. Although sterility testing is fundamentally a qualitative process, it's important to understand that precision still plays a vital role. When we talk about precision here, we're referring to how consistent the results are, despite some inherent variability. Specifically, this includes operator variability, which can differ from person to person, environmental condition variability, such as changes in temperature or humidity, and the reproducibility of contamination detection, meaning how reliably we can detect contamination across different tests. The approach we use to evaluate this precision involves having multiple analysts perform sterility tests on replicate samples. By doing this, we can observe whether the results are consistently positive or negative across different operators and tests. This helps us determine the reliability of our testing process. When we talk about precision in this context, we're referring to both repeatability and intermediate precision. Repeatability means getting consistent results when the same analyst performs the test multiple times under the same conditions. Intermediate precision extends that concept, examining whether different analysts, at different times, and perhaps under slightly varied conditions, still produce consistent results. Ultimately, assessing this kind of precision helps us ensure the robustness and reliability of our sterility testing process, which is crucial for maintaining product quality and compliance. Now, let's talk about the objective of this part of the study. We're aiming to assess the reliability of our method when small, deliberate variations are introduced. This is crucial because in real-world scenarios, minor changes can occur, and we need to ensure our method still produces consistent, dependable results. Typically, we look at parameters like a slight variation in incubation temperature, which might happen due to environmental factors. We also consider changes in membrane rinse volume, which can vary depending on handling. Filtration speed is another factor. Sometimes it might be a bit faster or slower than usual. Media lot variation is important too, since different batches of media can have subtle differences. And lastly, sample volume variation. Small differences in the amount of sample processed are also examined. The key outcome here is that, despite these minor changes, our method remains reliable. This demonstrates its robustness. That is, its ability to withstand small fluctuations without compromising accuracy or consistency. Highlighting this robustness is essential because it gives us confidence that the method is practical and dependable in everyday laboratory settings, supporting the overall theme of quality and reliability in our testing processes. Now, let's talk about the growth promotion test, or GPT, which is essential to verifying that our media can support microbial growth effectively. It's important to remember that this test must be performed for each lot of media and for every shipment we receive. This ensures consistency and reliability across all batches. During the test, we inoculate the media with specific organisms and observe whether they demonstrate growth within a defined time frame. The key point here is that the organisms must show growth within this expected period, confirming that the media is functioning properly. Overall, the GPT is a critical part of media qualification, helping us maintain quality control and ensure our testing environment is always reliable. Now, let's talk about sterility tests and the environment they require. These tests need a grade A laminar airflow hood, or LAF, combined with a grade B background environment. This setup is crucial because it ensures that the testing area is as sterile as possible, minimizing any risk of contamination. Qualification in this context means confirming that all these conditions are consistently met. This includes media fill simulation, which is a practical way to simulate the actual testing process and verify that aseptic techniques are effective. We also have personnel aseptic qualification, which ensures that everyone involved is properly trained and consistently performing the correct aseptic procedures. Environmental monitoring is another key aspect, where we regularly check the environment for any potential contaminants to maintain that sterile condition. And finally, gowning verification, which is about making sure that the proper gowning procedures are followed every time, creating a barrier against contamination. All of these steps are critical for maintaining a contamination-free environment, which is absolutely essential for reliable sterility testing. Remember, the integrity of the testing process depends heavily on both the environment and the operators, so qualification in these areas is fundamental to our overall quality assurance efforts. All right, let's dive into the key components of the validation protocol. First, we have the risk assessment, which can utilize tools like FMEA or HACCP to identify potential risks early on. This helps us prioritize our validation efforts effectively. Next, we need to record media GPT records, ensuring we have thorough documentation of our media preparation and testing processes. Then, there's the method suitability study data. This is crucial because it confirms that our methods are appropriate and reliable for our specific application. We also include the sterility test qualification summary, which summarizes the validation of our sterilization processes to ensure they meet required standards. The validation report ties everything together. It provides a comprehensive overview of our validation activities and results. Change control and revalidation criteria are equally important. They define when and how we should revisit our validation to accommodate changes, maintaining ongoing compliance. Finally, the validation documentation requirements specify what records and reports must be maintained. Overall, this protocol ensures that every step of our process is validated, controlled, and documented, aligning with our goal of maintaining high quality and safety standards throughout the life cycle of our product. All right, let's take a closer look at the acceptance criteria summarized in this table. First, specificity is crucial. We need to see growth of all challenge organisms, ensuring that our method can reliably detect them. Moving on to method suitability, we want to make sure that there's no inhibition, so recovery should be comparable to the control, confirming the method's effectiveness. Sensitivity, or our limit of detection, should be able to identify between 1 and 10 colony-forming units, which is quite a sensitive threshold. Robustness is also key. It means our detection ability shouldn't be impacted by minor variations in the process. Regarding GPT, growth should occur within the specified time, ensuring timely results. Lastly, environmental control must stay within grade A or B limits, maintaining the proper conditions for accurate testing. Overall, these criteria ensure our method is reliable, sensitive, and consistent, aligning with the overall goal of delivering accurate and dependable results. Let's take a moment to look at some common pitfalls and their solutions. First, product inhibition can be a challenge, but we can address this by optimizing dilution and using neutralizers to mitigate its effects. When it comes to media contamination, strict aseptic handling is crucial to prevent false positives and negatives. Ensuring a sterile environment helps maintain the integrity of our results. Operator technique issues are another key factor. Providing proper retraining and qualification for our staff ensures consistent and accurate handling. Environmental excursions can also impact our testing, so we need enhanced monitoring and prompt corrective actions to keep everything under control. Lastly, false positives or negatives often require root cause analysis, followed by corrective and preventive actions, CAPA, to identify and fix underlying problems. By understanding and proactively managing these pitfalls, we improve the reliability and robustness of our processes, which ties directly into our overall goal of maintaining high-quality standards. When it comes to regulatory expectations, it's essential that we adhere to full compliance with compendial methods unless we have a justified reason not to. This means following established standards to ensure consistency and reliability in our testing. Additionally, we need to validate that our methods are suitable for each specific product, making sure they accurately measure what they're supposed to without interference. It's also crucial to demonstrate that these methods are sensitive enough to detect small changes and are non-inhibitory, meaning they won't interfere with the product or the testing process itself. Lastly, maintaining aseptic assurance through proper qualification is vital to prevent contamination and ensure product safety. All of these points collectively help us meet regulatory expectations and uphold the integrity of our quality systems throughout the development and manufacturing process. Sterility testing is a critical safety measure in our industry. It's essential to ensure that our products are free from microbial contamination before they reach patients. Method validation plays a key role here. It guarantees that our testing methods are reliable, accurate, and compliant with regulatory standards. When we have a solid validation strategy in place, it helps us minimize false outcomes, whether they are false positives or negatives, and ultimately enhances the safety of the products we deliver. That's why partnering with experts like Siphacos Consulting can make a real difference. They support us with end-to-end microbiology validation services, ensuring that every step is handled thoroughly. In conclusion, rigorous validation and expert support are vital to maintaining the highest safety standards and building trust in our products. For further support, please feel free to contact us. We're here to help you with any questions or additional information you might need. Thank you so much for your attention and engagement throughout this presentation. Remember, we're committed to assisting you whenever you need it, so don't hesitate to reach out. Thank you again for your time and participation.